So I want to spend a little bit of time this morning talking about this time of year as folks start running their 360 SOILSCAN machines, and sometimes they'll send parallel samples off and go, gosh, the results are different. What's going on? Why am I seeing what I'm seeing? Does that mean somebody's right and somebody's wrong? And so just want to kind of review that a little bit and talk about some of the things we're seeing and answer some of these questions like, hey, why are my results different from a lab? And again, what does that mean? So before I do that, I want to go back real quick and just discuss with you nitrogen in the soil. Again, why might a lab result be different from my SOILSCAN result? Well, you got to think about, and here's what I want to talk about it. We pull those samples, all right, and those samples have ammonium nitrogen in them. Because think about it, if we're using anhydrous or manure, 28% is basically 75% ammonium fertility, and urea is going to be 100% ammonium fertility. So those soil samples have a lot of ammonium in them, right? And that ammonium, once you mix oxygen in it and you warm it up, okay, it converts and becomes nitrates. And that's, of course, what we're trying to test with SOILSCAN. And that's what you will send it into a lab and you'll test and you'll say, okay, hey, I want to test for these products, and that's what you'll pick up. But keep in mind, as we warm those soils up and those samples have ammonium in them, we can convert some of that ammonium into nitrate during a handling process. And so I just want to share a little bit about what we've found. If you go to the next slide, Caitlin, we've done some work, and they're really, I'm sorry, let me bounce this real quick. There really are two parts I want to discuss as we think about the process. Is there conversion that goes on in the handling process of getting that sample to the lab? And then also just want to talk a little bit about what happens to that sample in the lab. What do they do with that sample, and could that impact it as well? So, and again, I guess I want to start with this perspective. We're not saying anybody's right or anybody's wrong. It's just different, and we want to talk about the differences and why that might show up in a testing environment. So let's go. And what we did here, this is some interesting work. I'm going to show you 3 slides. And what we did is we took samples. Now, when I say we, I think this is Greg Urey and Jason Keenest in Southern Illinois. And they took the same sample and split it, okay? On the right 2 columns, all right, what they did is they froze the sample and put it in the mail, which is what hopefully many of you do, right? That's the standard practice of freezing it and putting it in the mail. But then what we did was we took that same sample, froze it, but instead of shipping it in normal, just throwing it in a box and shipping it to UPS, we put it in a cooler, packed it to keep it really, really cold. And when this one lab sent the results back, here are the differences we got. And I'll be really candid with you, I was really surprised. Back to the, my axiom about every day I live, I learn how dumb I was the day before. I was really surprised by these results. But basically, where we shipped froze the sample and just shipped it UPS standard, okay, the nitrate results, again, same sample, same lab, were almost 2x. And interestingly, if you look at the third line, so there was a fall anhydrous, a spring anhydrous, same rates, and then manure, right? It was accentuated even manure in that sample. So think about what we talk about handling, and I will tell you, I've talked to folks, and a lot of this discussion is we don't think they change that much in shipping. But I would tell you, we just got these results back. We're going to continue to repeat these tests, but I was really surprised by this result. Now let's go to the next slide, Caitlin. And by the way, this is, if you take those same numbers and you convert them to pounds, okay, that's what those differences look like in pounds. And look at where we had that manure, okay, that's those numbers converted to pounds. It was pretty surprising that the differences, again, same sample, the only difference here, same lab, is one sample was frozen and shipped standard, the other was frozen and shipped in a cooler to keep it cool. And there's the differences we found. Now, what we also did with that same sample, and I want you to focus on the 2 right columns, okay? Because what we did is we took that same sample, okay, and we shipped it to 2 different labs. So lab 1 is the center columns, and lab 2 is a column on the right, the 2 columns on the right, okay? So again, now we're not talking about frozen and cooler versus frozen and UPS. Both these samples were frozen and then shipped standard to 2 different labs. And here's the differences we got. And here I think is the really important point as we thought about why they might be different from this lab. Lab number 2 is about a 1-day ship time, okay? Lab number 1 is about 300 miles away. So we're pretty confident that those results in the center, all right, sat in the mail longer, sat in a box. Again, they were frozen and then just shipped standard. And we're curious if the differences we're seeing from lab 1 and lab 2 could have something to do with what's going on in the shipping time in the box. And again, these were frozen before they were shipped. Imagine if we just pulled the samples, put them in a bag, put them in the mail, and shipped them that way. So again, as we think about comparing a soil scan to a lab, all right, here's what you do with SOILSCAN, right? You go to the field, you pull the sample, and you immediately, hopefully, within a few hours, you're testing that sample, right? So in theory, if we test it pretty quickly, there's not a lot of the conversion of that ammonium nitrogen over to nitrate nitrogen. And keep in mind, we are using water to access and flush the soil, right, of those nitrates. So that's the process you use with the SOILSCAN. Compare that with a soil lab, right? We pull the sample, maybe we freeze it, maybe we don't, maybe we ship it in a cooler, maybe we don't, but it sits in the mail. And I would tell you, when I just asked someone who runs a soil lab and said, hey, how many of the samples do you get that aren't frozen when you get them? And the answer was many. Quite a few of the samples aren't really frozen, okay? But then the other thing that occurs, right, is that lab takes that soil, grinds it up to really, really fine, which is good. And again, I want to make sure you understand. I'm not saying this is wrong. It's just different, right? And then they slowly warm it to 104 degrees. They use 104-degree temperature to dry it out. And the reason doing that, right, is to stop the nitrification process. They want to dry it so that really will stop the nitrification process. And that's good. That's right. But what we're wondering is if in some cases, that slow warming actually drives the nitrification in the sample. I'm not going to sit here and say, oh yeah, that's absolutely changing the sample, but it makes you think and wonder as you start looking at some data, could that be part of the process? And again, not bad, just different. It's the standard benchmark process that's been used. The other thing to remember then also about labs, there's different extraction techniques. Some will use like a cadmium extraction, but they'll use a chemical extraction to physically pull all the nitrates out of the soil, whereas we're using water, which again, that's what the plant is experiencing in the soil. So we use water, they use a chemical extraction. So I would expect there to be differences simply from the extraction method as well. So where do we see the biggest differences when we compare a soil scan to a lab? Well, I guess I want to start and say, right, that many times we don't see differences. They're very, very similar, and that makes sense. And I'll kind of review that a little bit here in a minute, but they're similar, and that's good. But we would typically expect to see some differences for a number of reasons. Again, as I mentioned, they take their sample, they grind it, okay, and they use a chemical extraction. That's a different process, all right? The other thing I would tell you is, and I'll show you some data here in just a minute of another test we did, is that it seems like the spreads tend to be a little bit larger when the samples contain more ammonium nitrogen, like soon after application or in a manured field. Why would we see that? Well, if you think back to the process we talked about early on, the nitrification process, if there's more in to convert, right, in that, say, the handling or sitting in the mail in a box, or maybe the warming process, if there's more to convert, it would make sense then that the spreads might be larger. Now, the one thing to keep in mind again, as we get closer to side dress time, okay, one of the things we think is once the majority of that ammonium is converted to nitrate, the numbers typically become a little bit closer together. You don't see those big spreads. And so by side dress time, much of it should be converted. So I want to stop there for a second. Just make sure that makes sense. When you have samples, and again, I'll show you some data where they're manured or you just recently applied ammonium fertilizers, right? That's where we can see bigger differences. And that makes sense to me because as, again, that soil warms and it gets oxygen, that would convert. But if you don't have a whole lot of ammonium nutrition in the soil, like close to side dress time, the differences in the spreads probably shouldn't be quite as dramatic. We did some testing to check on this, okay? And here's what we did. I have 2 slides I want to share with you. So what we did this time, okay, in the previous data I showed you, we were really talking about handling. Now what we did with this is we were talking about process and trying to find out, are the processes different? So what we did is we took a sample, all right, and that sample is the wet 360. In other words, we tested it just like we normally do with the SOILSCAN machine. We pulled the sample, ran it through the machine, got the results. Then what we did, and I'll show you another set of data, okay, we took that same sample and basically handled it as you would handle it in a lab environment. And by that I mean, what we did is we ground the sample up, we slowly warmed it to 104 degrees, and we started testing the results that we got. Interestingly enough, and QC, by the way, means quality control check. But interestingly enough, if you look at all these comparisons, the prior slide and this slide, 100% of the time, right, 100% of the time, when we processed those samples, in other words, ground them up, warmed them slowly, all right, the differences showed up in those samples. The other thing I'll draw your attention to is if you look at these numbers, kind of what we were talking about, where there's more nitrogen available to convert and nitrify, those spreads are bigger, right? They're larger. But where there's not nearly as much nitrogen, the spreads are smaller, and you don't really see those big differences. So let's advance on. So again, what I want you to understand is this is the same sample, and all we did was we ground it, warmed it slowly, dried it 104 degrees. And so I've shown you 2 sets of data. One was kind of what happens when we handle it, and then 2 is what happens when we process it, kind of like a lab. Does that affect it? Okay. So the question is then, okay, is the testing procedure bad? Is the machine bad? I'll talk about that in a second. And the answer is no, neither of those are true. Neither of them are wrong. We'll hit on that. But the one question I get a lot is, well, okay, now I don't understand, because you show us all this data from the Ag Laboratory Proficiency Program that says, you know, you're spot on, you know, that the SOILSCAN machine is measuring right. Well, how can that be if you're showing me data that says it's different? Well, let's think about the process for that ALP certification process. What they do, right, is they take a sample, a known sample, and they dry it. And when they do that, that dries and stops the nitrification. Then they send that sample out to the lab. So is nitrification occurring in the shipping of that sample? Probably not, because it's been dried and handled. Then the labs run their tests, and we run our tests, and as a result, it is a completely different process. And that's why, if you look at the ALP data, and I'll show you some of that in a minute, because they send out samples for testing that in essence have stopped any nitrification, any conversion. That's why we do so well in the ALP testing, because those are samples that are treated completely different. Whereas you think about the samples that get sent and compared from a lab, all right, that's a whole different process. So really it makes perfect sense to me in that why we pass ALP so well, so thoroughly, is because it's a completely different process. They send out samples, known samples that have dried, stop that nitrification process. And I just wanted to touch on that again real quick. We've got 3 years of data, and I just wanted to share, just to remind you that if you look, that left column is the SOILSCAN, that's 2014. And then they send this out and compare it to a number of labs. I believe it's 8 labs, and right, you can correct me if I'm wrong, it's 7 or 8 labs. And then the center is the median of all the results. And you can see in 2014 and 2015, all right, we do really, really well. And those are basically, if you think about it, these are apple-to-apple comparisons where the sample is handled, the nitrification has been stopped, and so it makes sense that we do well. Can you go to the 2016 results that just came in? Next slide. There we go. So these results just came in as well, and it's 2 different testing methods. But again, if you look, the SOILSCAN machine is that left column, and then the median for all the labs is that center column. You can see that the SOILSCAN machine does great. I mean, I guess I would call that an A+ grade. and it's doing what we thought it would do. So I guess the question, are we saying that a soil scan is correct and the labs are wrong? No, that's not what we're saying. We're saying that both are correct, just different. In other words, the numbers that a soil lab gives you, those are the right numbers, right? Those are the correct numbers for the sample that they get, and the soil scan numbers are the correct numbers when you run those samples as well, okay? They're just different because the samples are handled differently. So the question then is, well, how do I explain that to my customer, right? I'm not even sure I get it, but how do I explain it to the customer? And here's what I'd say. As part of our system, the complete 360 system, right, we've taken the results of the soil scan machine, we combine it with our nitrogen app, the nitrogen calculator app, tested that, and I'll show you some data in a minute. And the bottom line is we have complete confidence in the combination of your 360 soil scan with the nitrogen calculator app to give you good recommendations. Again, it's our system, right? It's a little bit different, but keep in mind that we've tested this, and what we find is our system, okay, of testing it right at the time of the application, then making your calculation, is working great. Okay, so again, both ways are correct, just different. Go to the next slide. Some of you have probably seen this data, but we spent a lot of time comparing the soil scan system, okay, to other systems that are out there. And those green bars, okay, are the soil scan. The gray bars are basically a couple different models. So is the red bar. There's 3 different models. And then the blue bar is frankly a really top shelf agronomist, and we compared it to his recommendations. And I guess what I want you to understand and take away from this slide is that our system, the 360 soil scan in combination with the calculator app, it is something that's been tested, and it works. And so we have really good confidence in the technology. We have really good confidence in the information. We have really high confidence in the system that the decisions you're making based on our system are good, solid agronomic system decisions. Here's the bottom line, I guess, and that is that we're not at all surprised when lab results differ from 360 soil scan. Our own data would confirm that point. I will say, again, I'm surprised how much, really, and we're going to keep testing this and trying this, and I'll freely say we've taken some samples and we've thrown them in a truck and let them sit there all day and test them, and we don't see a lot of differences. So nitrogen it's really a slippery pig, and sometimes it's hard to get a complete understanding. We continue to gain understanding, but again, I guess I want you to understand that we have real confidence in the REX and the system that you are using with us, and it's okay to have confidence in the soil lab results because their results are good too. It's accurate, okay? It's different because frankly it's a different system. The other thing is, keep in mind, this is a great tool to help ground truth some of those nitrogen models. There's a lot of models coming out, and I would continue to stress to your neighbors, your friends, that it's a great tool to help you ground truth those nitrogen models. And again, I wish we'll wrap it up with this point, saying neither system is wrong, okay? The soil lab numbers, those are good numbers, but we just have to understand that we've worked with our system to develop these recommendations, and we're confident they're going to work on your farm. So, all right. I'm going to stop. I don't even know if I have any more slides. I don't think I do. But John or somebody, are there any questions that are popping up? And Ron, Caitlin, if you'll unmute Ron Lloyd real quick, I'd be curious if Ron has any comments to share with the group as well.
Yeah, great job. The only thing I'd add is I believe that the confidence that we have in the soil scan machine is when we're taking that fresh right in the field, we're taking out that variability of handling. And that's where I think that difference, like you said, and I agree, it's not that the tools at either the lab or the soil scan are bad or wrong. They're getting the right reading for the soil that they're testing. But when we test in the field fresh, before we've allowed it to mineralize and do things unnaturally, it goes back to what Matt talked about. Everything in biological systems revolves around oxygen and around moisture and around temperature. And when we're mixing that sample really well, and it has some moisture to it, and we're introducing oxygen, and it has that temperature during that shipping process or sitting in the truck, we are introducing some unnatural mineralization. And I think that's why, we have the confidence we have of taking that fresh, because what I really want to see is, what is the corn plant seeing? What is the environment that corn plant, the corn roots, what do they have access to? And if we alter that soil unintentionally, we could get numbers back, like you showed in some of your previous slides, that maybe don't represent what the corn plant's actually seeing. So that's the only part I would add, Jim. But I thought you walked through that very well.
Ron Lloyd, there's a question, and you've got experience with this that came up. Do we have a suggestion for a soil grinder, or do we think it's not needed? And Rhett, you can jump in as well. But do we have any suggestions to grind the soil, or is it not necessary?
Yeah, great question. Typical me, it depends. When I'm in good, what I'll call, good silty soils, and when you take it, if you take a sample and you put it in that bucket and it just crumbles, and you can get a nice, homogeneous mix that we talk about a lot, no, you don't need to grind it. But a lot of these soils, if they're clay or they're a little bit dry or a little bit wet, they can be more difficult to really get that soil mixed evenly. And you need to just look at that sample well and think about it. If I'm just taking 2 random scoops out of that sample, do I feel comfortable no matter what 2 scoops I took out of that sample, they're going to be the same? And if they're not, then yeah, I would highly suggest grinding it, mixing it. We do several different ways here in our operation with Jason Keenest and Zach Wiegand and our interns and agronomists. What we typically do is, if you're on a smaller scale, you can get a blender from Walmart, a pretty cheap blender for $20. It'll work pretty well, whether it's a Ninja brand or whatever. The thing we've done, because we do such a high volume of samples, we've actually taken those kinds of blades and mounted them on a metal rod and put that in our drill, in our battery-powered drill, and we'll mix them up. And that's when it gets tough. That's only when we run into those tough conditions, because these guys are out every week taking samples. So, when we're on a schedule, it really doesn't matter what the condition of the soil is. We're going to do it because we want to build that database. So we will run into tough conditions. So again, just look at that sample if you can do it properly. I did some the other day at home that were good, and I was able to do it with my hand. And there was some others, it was a little wetter, and I actually used a home mixer and did it that way. So that's my opinion, been my experience. But again, it's just looking at that sample and making sure that you feel comfortable it's well mixed.
I would just follow up that with the new sensor that we've got out there, we've seen some really good results that I put up against any lab in the country. Use it, SOILSCAN, for what it is. You're trying to determine what your application should be. So, going out there now is interesting. It'll open your eyes to what Mother Nature is doing and how your soil is changing. But more importantly, when you're going out to do that side dress application, go out there, and SOILSCAN is a great tool to tell you if your tank's empty, quarter full, half full, or full. That's really the bottom line and the power of SOILSCAN and what we developed it for.
All right, thanks everybody. You take care of yourself, be safe, and we will talk to you in a couple weeks.